Hey, what's going on? Are you okay?
I'm just feeling stressed and overwhelmed about running my assay. There's not enough in the budget, and it takes forever to get the results. My lab assistant is exhausted from waiting for results, but I don't have a choice. I'm gonna have to cut back another lab project.
Whoa, what are you talking about? Have you not heard of the ATAC sequencing method? It allows for complete genome-wide mapping of chromatin in less time and is way more cost-efficient than other methods.
What? I've never heard of it. Who came up with this method? This is madness.
Well, ATAC-sequencing stands for Assay for Transposase Accessible Chromatin Using Sequencing. The method was first published in 2013 by Jason Buenestra. The lead researcher wanted to overcome the limitations of current methods used to study the nucleosomes of the accessible DNA.
Okay, but what exactly is ATAC sequencing?
Well, ATAC sequencing allows for genome-wide mapping of chromatin. Unlike traditional methods, it does not require prior knowledge of regulatory elements, and it's better to study transcription factor binding, gene regulation in complex diseases, T-cell activation, embryonic development, and even cancers. When compared to other similar techniques such as RNA sequencing and chip sequencing, we can see that ATAC sequencing provides certain benefits that neither of the other techniques can offer. One similar technique, as mentioned, is called RNA sequencing. RNA sequencing provides information on the differential expression of genes, whereas ATAC sequencing gives a more in-depth explanation on how gene expression is controlled with regards to the open accessible regions of DNA in the genome. The other similar technique, called chip sequencing, is a common method for epigenetic mapping that focuses on protein-bound DNA sections, which needs approximately 1 to 5 million cells, compared to ATAC sequencing that only needs 25,000 to 75,000. By needing a lot less cells, ATAC sequencing allows for unique cells and cell subtypes to be analyzed, compared to the other techniques where these unique cells, due to their small quantity, could not be analyzed. ATAC sequencing also takes around half the time necessary for chip sequencing assays and thus results in a lower cost. This puts into perspective how using ATAC sequencing can actually save users money, starting materials, time, and overall yield results that previous techniques could not. It further opens up the door to combine sequencing techniques, if desired, to generate a larger range of information on the cells being studied.
Now that I can see how important it is, how does ATAC sequencing work?
Well, I'll let my assistants explain.
For sure.
Well, in ATAC sequencing, there are two simple steps. The first step involves collecting a cell sample. It is important to count the cells for the transposition reaction. Otherwise, it could lead to the improper digestion. The goal of this step is to take intact cells and lyse them in order to generate pure nuclei. The range of cells to gather is 25,000 to 75,000 cells.
In the second step, the resulting chromatin is broken up and simultaneously put together with sequencing adapters using TN5. TN5 is a transposase which allows for a cut and paste method for accessible DNA. Sequencing adapters are then inserted to the chromatin resulting from the transposase activity. Finally, the tagged DNA fragments are amplified using PCR and barcoded primers to yield an ATAC sequence library. The library can then be analyzed by qPCR or next-generation sequencing. This will further help us gain insight about the source of many different diseases and cell types.
Wow, that's so much simpler. Now I don't have to stress over mapping nucleosomes and accessible chromatin regions. Can you tell me more about its applications?
Of course, ATAC sequencing allows for the assessment of accessible chromatin. Therefore, it can help study the role of gene regulation in many diseases. This method unlocks the ability to look at regulatory mechanisms in certain diseases and discover certain biomarkers. Also, when combined with other techniques for gene expression, it can expand the library of information of the cells.
Oh my gosh, thank you for telling me about this new and innovative technique. My lab will no longer be frustrated while having to wait for our assays to run. We can accomplish more in less time, and we won't have to break the bank.
No problem. Everyone should know about ATAC sequencing and its applications.